Because cancer has hurt the lives of millions of individuals, for my project this year, I wanted to find a way to inhibit survivin in melanocytes using siRNA. siRNA is a type of RNA interference that stops gene expression. Survivin is part of the inhibitor of apoptosis protein family. So by using siRNA to inhibit survivin, I can therefore make cancer cells commit cell suicide. Specifically, what this project is trying to do is create individualized treatment options. Because cancer is so diverse, we need to allow for the specificity of treatments, and siRNA allows for this specificity. After doing some research on this topic, I hypothesized that the method of siRNA transfection to get the siRNA inside the cell, Bradford protein analyses to measure total protein content, and Western blots to measure specific protein content would give me data that I could use to determine whether or not I had inhibited survivin in melanocytes using siRNA. According to the results from the data of my control experiment using GAPDH, I found that in well number 17, there was the highest knockdown with the least toxicity of GAPDH. This well had reagent 4 at its highest concentration. So therefore, I used reagent 4 at its highest concentration in my experiment with survivin. In my experiment with survivin, my data shows that the highest knockdown of survivin occurred within 72 hours of exposure to siRNA. Therefore, I can conclude that my hypothesis was correct, and the method of transfections, Bradford protein analyses, and Western blots can work to inhibit survivin melanocytes using siRNA. This project widens the range of treatment options that can be given to patients with melanoma. And because siRNA specifically targets a certain gene, in this case BARC5, it can therefore reduce the potential for toxic side effects that are associated with current chemotherapy methods. Thank you.